Okay, good. Like, for example, muscle cell. And the RNA is the transcription. Remember, we go from RNA, DNA, to RNA, which is single-stranded. That RNA then is ferried out of the nucleus and into the cytoplasm, where it connects with that thing called the ribosome. And then that code, genetic code, now in RNA instead of DNA, is then translated into amino acids. So for each 3 letters, there's 1 amino acid. There are some that are redundant. And in terms of gene expression, it's not exactly an on-off switch, as we'll see. So, like for example, muscle cell. There's these RNA transcripts, and then there's this protein that is translated into, and a certain amount of the protein. And then there's also transcript for protein B, less amount of it. C, not as much as A. Then gene is off. For a you actually might have the same genes turned on, like housekeeping genes. In terms of okay, gene B is turned off. In other words, it's not expressed. C is expressed, but a different amount. D is also expressed, unlike the muscle cell. In the nucleus, do we just have the genes that cell needs, or do we have the whole genome? Anybody? Like in the nucleus of each cell. What do you think? Okay, I'm going to ask someone. What about you in the back? What? Okay. In each cell, in the nucleus, in terms of genes and chromosomes, is it just the genes that cell needs, or is it the whole genome? Yeah, exactly. But remember, each cell that we have has the whole genome, all 3 billion base pairs, 19,000 genes, but only some of those are expressed in some cells versus others. Okay, and we went over this before. There's oftentimes promoters, regions, in other words, just upstream from where the gene is on the chromosome, that actually regulates how much of that gene is expressed. And there's also these enhancers that can be all over the whole genome. Like you could have something on another chromosome that still regulates what's going on this original chromosome that we're talking about, where that gene is found. So, does the promoter region act as an on-off switch? No. Instead of an on-off, it's more like a dimmer. There, like that, right? So now we're saying that the amount of product, protein product, that's actually produced is like a dimmer. You can make more of it, you can make less of it. It's not necessarily on-off. Okay. Okay. Now, the part of the gene that actually codes for amino acids in a protein is called the coding region. The promoter and enhancer regions are just for gene expression. Many times, it's not the structural genes that you might find mutations in. A lot of times, structural genes like lactase. With lactase, young mammals, baby mammals, can drink milk and get glucose from that. And then, when they're weaned and they no longer get milk from mom, they're never going to see milk again. And that gene is then downregulated, basically turned off. But as we know, some human beings are lactose tolerant. It's happened 3 individual times with 3 different populations, including the Middle East, and then that was then brought into Europe, then 2 different groups of populations in Africa, where the lactase, which is the enzyme that breaks down lactose, was kept on. In other words, the promoter region, instead of stopping the production of lactase, and you can imagine that you're never going to get lactose again in your diet, making an enzyme you're never going to use is going to be expensive, and selection is going to remove it. But in these populations that are lactose tolerant, some individual had a mutation, and it turns out the mutation was actually in the promoter region. And each of these 3 different populations, it was a different mutation, but in the promoter region. So each of those mutations were slightly different in terms of what they coded for in the promoter, but they kept the gene on being expressed and producing lactase. Okay. Now, gene expression is actually, understanding it is rather new. There are regulatory elements active in different tissues, as we know. Regulatory elements are often located far from the genes they control. There's a network of regulatory interactions unique for each tissue. There are also ways to shut off genes from being expressed by attaching what are called methyl groups. This is called epigenetic genetic control, and we're now understanding it much better. Oftentimes with cancer, it's changes in the epigenetic or regulatory genome that is actually, there's a mutation there. Okay? So you can see all of these different, these are all different amino acids. How many letters does it take to make one amino acid? How many? 3, exactly. That's called codon. So you might have TTA, and that will correspond to one amino acid. So you can actually see how all these regulatory elements, including promoter regions, have many different attachments. And it's a very involved, complex process in terms of gene expression. Some more. So it's kind of like what we discovered, eating disorders. It was pretty much binge eating and drinking the whole time. I guess I better call. This is actually serious. But anyway, that's what I did. So those of you who just sat around watching TV, there's more to life than school and TV. You may not know it, or games on the internet or whatever. There's actually stuff out there. I just want you to realize that. We have an exam. I know you're all excited, right? Thursday, brain scantrons. It's going to be the same sort of format, 50 questions, plus some extra credit questions as well. And there's a study guide. We'll go over a little bit of that, since we probably have a lot of time. But first, I just want to say a few things. Okay. There's a video on this, and then okay. Do cows and, okay. Remember this bovine growth hormone is injected into cows. They make it, but you can inject more into it and they'll make more milk. To get more made of it, the gene for it is actually put into a bacteria. So, do the cows in this example qualify as a genetically modified organism? Was the cow genetically modified or was it bacteria? Bacteria. So I just want you to make sure you understand which is the transgenic organism. Because there's going to be, on the test, there's going to be a question, I assure you, just like this. Okay? Okay. Now, as we know, as I've talked about earlier, we use genetic modification in all sorts of different ways, including in medicine and research, as well as in agricultural products, whether it's plants or animals. Now, in a sense, humans have been doing this for a very long time. It's called selective breeding or artificial selection. So, like for corn, okay, there's the original ancestor of corn. It's called teosinte. It's found in the Mexican highlands in Mexico, and it was domesticated, and it was basically selected for bigger and bigger kernels until nowadays we have corn like that. This is called selective breeding. Same thing happened with wheat. Same thing happened with some animals, like our cows are actually descended from a wild, they were called oryx, and they were domesticated twice independently, once in Central Asia and Europe, and the other in India. So like Brahma bulls with big hump, those are Indian. They're very good in tropical climates. So they were selected for various things. Now, in terms of genetically modified foods, we now do something different. So in the past, with selective breeding, you'd want a certain trait. In this case, let's say, these large kernels. The trouble is, when you're selecting one trait, some traits are correlated, and it means essentially that when you change one trait, you drag along other traits with it. And a good example of this is, if you look at dogs, they're descended from wolves. Some dogs look a lot like wolves and others don't. There's some big and some small. They're bred for different things. A lap dog is just to sit on your lap. A border collie is for herding. Animals like goats, sheep, things like that. Terrier, for chasing rats. So there's all the selective breeding going on. Now, in the process of selecting for these domesticated traits, one of the things, of course, that was selected for, whether consciously or not, was personality type. In other words, you don't want an animal that is aggressive towards humans or scared of humans, like wild animals are. And so, originally, the first domesticated dogs, they were selected for tameness. In other words, they're not terribly scared of humans, they're used to humans, they're trained by humans, that sort of stuff. Well, when that happened, a bunch of things happened, one of which was they, instead of getting that kind of pointy face like a wolf, they got a lot of them got a more rounded face, like a retriever. Their ears flopped. They got changes in coat color. Their tail, instead of being straight, oftentimes turned up curled. These are all traits that were dragged along in terms of selecting for a changed personality. They actually did the same sort of thing in Russia with silver foxes for the fur trade. And the guy that did that, it was discovered where he did these breeding experiments and selected over generations for tame foxes. And the same sorts of changes happen. They got floppy ears, changes in coat color, so they became, of course, much more tame. All these other traits went with it. Well, with selective breeding, that's what happens. But with genetic modification, you can actually just insert just the genes and the traits that you want into a genome. Whether it is from another organism or another breed, something like that. Okay. Now, as I mentioned in agriculture, there's been a lot of genetic modification. So, a lot of people on this planet, their main sustenance is rice, especially in Asia, Southeast Asia. And a lot of them eat white rice, which has a lot of calories, but it doesn't necessarily have all the other nutrients that you actually need. Certain vitamins and stuff like that. So some of these human populations, they have to get their minerals and vitamins from other sources. But what they've done with this rice is they've actually inserted a gene from another organism that makes something called beta-carotene. And beta-carotene is actually a type of vitamin A, and this gene is expressed in this rice that people are growing, and it's called golden rice. And so it's being disseminated among all these different farmers, so that when they grow rice, they're actually growing rice with vitamin A in it. And that's a good thing. So there's a lot of people who are worried about GMOs. Essentially, in fact, many European countries actually ban GMOs. They think perhaps that they're not the same, they're not natural. Well, they are in many senses. Now, doesn't mean that they're always good. Like in a lot of genetically modified plants, they've inserted a gene that makes the plant resistant to the most commonly used herbicide called Roundup, glyphosate, and this is sprayed on fields to kill all the weeds. But if a plant has this gene, it means that you can spray it and it won't die. It's immune to it. Okay. The trouble is that plants especially, there's often a lot of gene flow among different species. Animals not so much, but certainly some plants. In other words, their pollen can escape and maybe they can actually pollinate another species. And carry that allele that makes them immune to Roundup into a wild plant. And you can imagine how that might actually wreak havoc on the ecosystem. You suddenly have a wild plant that is resistant to Roundup, and it spreads exponentially all over the place. So, now, one thought for this was that you can actually put in a gene that makes the next generation sterile. So you buy these seeds that are genetically modified from a certain company, you plant them, they grow the plant, but those plants can't produce new seeds. But they're not really doing that, and so there is a problem with this idea of genetically modified genes and alleles being spread. For a while, You can actually introduce these new alleles with what's called a gene gun. They don't really do that much anymore. Where you can actually take these stretches of DNA and kind of shoot it in, put cells in a whole bunch of places. They start expressing that protein. The same thing has happened elsewhere, but the newest thing that's going on, there's been a breakthrough. It's called CRISPR. And what CRISPR is, I'm not going to tell you what it stands for, but it turns out that bacteria are often infected by viruses. So they actually have a system where they remember viral sequences, or those viral sequences are copied into this CRISPR system so that when they get reinfected and it's recognized, then it goes in and cuts out these viruses. Okay? So this is what CRISPR is. It turns out we can actually modify this and then use it to insert genes or take out alleles in sequences a lot easier. Before it was a hit and miss sort of thing with like transgenic animals and plants and everything. Well, now it can be very precise. So this is kind of what's going on now. You can, let's say, target certain genes in here, and you can insert new genes or take them out and repair it. And so you have targeted genome editing, and they've done it for all sorts of things, including human cells, zebrafish, bacteria, plants, all that kind of thing. Okay. Now, like I mentioned before, there were transgenic organisms. There was still selective breeding. So people would plant these crops that were pretty much genetically identical. Clones, like a whole field of corn. And on the left, this might be selective breeding corn, or it might be genetically modified corn, but there's very little variation in it. And, which is fine, but let's say there's a new disease that invades, and these plants are susceptible, it would wipe out all the corn. And this is exactly what's happened with bananas. The banana that most of you are familiar with is called Cavendish. It was developed by selective breeding. It travels well. It doesn't bruise easily. So you can pick it green, it'll ripen, and then finally you can sell it later on. You can move it all over the place. Deliver it to, let's say, North America, because they're tropical, or deliver it to Europe or whatever, and that's great. The thing is, Cavendish are now susceptible to this fungus, and that variety is being wiped out. So if a plantation of bananas of Cavendish get infected, the whole plantation has to be cut down. It's finished. So they're now trying to come up with a new variety that's resistant to this fungus. Now, in nature, we have a lot of different variety going on, especially in terms of reproduction, sexual reproduction. You can spread alleles throughout the population and let selection act on it. But if you don't have the variation to begin with, this is the problem that can happen with genetically modified crops or just, domesticated products, you get a lack of diversity. So, like the Cavendish, they're coming up. Now, it turns out there's many other different kinds of varieties of bananas. Anybody in here ever eat plantains? Yeah. So, especially in Caribbean countries, they eat these kinds of bananas that you actually fry up. They're delicious if you've never tried them. But there's all these different varieties out there. The thing is, a lot of those varieties don't travel very well. So that's why they're developing new ones as we speak. And they're also going to try to perhaps put a gene from some other type of banana into Cavendish so that it can actually resist the fungus. But they haven't accomplished that yet.